hearing a lot about. This large group of people has worked very hard to create this, so I want to thank them. And then off to the right are program colleagues of mine from National Human Genome Research. Elise Feingold was at the beginning of the project and started it out, and Dan Gilchrist, my colleague, is here with this meeting today. So I'm going to tell you a little bit about the ENCODE resource, what's the rationale for it, what is it, how is it made, a little bit about how it can be used to study human disease. That's not all that it can be used for, but that's one important application, and a little bit about how to access ENCODE materials. As the ENCODE makes me think in some ways of music, it has collections of lots of different things. They all work together. There's not the one thing that matters or the one thing that's important. Depending on what you're doing, you may care more about one instrument or another, or what kind of music. The same kind of thing takes place in ENCODE. So the reason for ENCODE, I think, is quite clear. At the completion of the Human Genome Project, it became obvious that understanding much of the genome was difficult. Understanding what portions of the genome are doing something of value is difficult. We have a genetic code that tells us about protein coding regions. This is a very small part of the genome that's very sparse, right? So how do we find the regulatory regions? How do we find out what they're doing? And how to identify them is another problem. And this is important because we know that non-coding DNA does a lot of important things. And heritability of the associations is imputed to lie in non-coding regions, so that's important. There are Mendelian disorders that are caused by genetic variation in non-coding regions. So by those two measures, among others, non-coding DNA is important. But it's not easy to study, and this functional information could be used to interpret disease, gene regulation, mechanisms of mutation, and so forth, if we had a better understanding of it. So ENCODE is one project. A lot of individual investigators are also working in this area, trying to get a better understanding of these issues. Here's a graphic way to see the problem. You just take a snippet of the genome, not a random snippet, but snippet. Looking at it quickly, it's difficult for most people to get information out of it. You know, to some extent, this is an exaggeration, right? We all know that partially this is a solved problem. You can have apps like at the bottom of the slide where you can mark exons and you can mark variants that are associated with disease, or you can do the same thing by color coding the sequence. And that gives you some information now that you didn't get just from looking at the sequence. But I would argue that this is still pretty sparse in terms of information content, even though this is very valuable. So the approach that ENCODE is taking, a lot of individual investigators are doing this as well, is to build very richer maps so that we might have more information. So how this might work is now you can see the same GWAS variant in context of the gene. This comes from non-ENCODE work. And from ENCODE data, you can see that there are biochemical signatures in this region. We'll explain what they might look like. And it turns out that this is the signature for a candidate regulatory region. So that tells you something about this variant could lie in a regulatory region. ENCODE data can tell you other stuff because it's cell type specific. You can see that this information is preferentially coming from a particular cell type. So that may give you some idea of what cell type is important in this process. And while this variant appears to lie within a particular gene, you can see that easily from sequence alignment. There are ways to predict what gene it might be worked with. This is, of course, predictive, not known with certainty. And in this case, the prediction is that if it's a regulatory variant, it's actually working to control the neighboring genes. So being able to have these stories moving forward would help us much better appreciate the role of genetic variants and phenotypic traits. So the goals of ENCODE are to state how to get to this story. We're trying to identify all of the candidate functional elements of the genome. We're also trying to share this resource freely with the community. Freely means it's unrestricted access, not controlled access. There's no login, there's no purchase. You simply go to the website, use whatever it is that you want. And people are using this to look at the genetic basis of disease, gene regulation, how mutation occurs, and so forth. But any process that you'd like to use it for is allowed. So the way ENCODE is doing this resource is built upon decades of research in gene regulation. For years, people have known different biochemical signatures are associated with gene regulation, transcription, post-transcriptional processes. And these have been converted into genome-wide assets. And then the results of these are used to sort of reverse engineer to make predictions about how the genome is working. So you can make predictions about where genes are. It's probably the simplest part. What are the transcripts that are encoded within those genes? And then what are regulatory elements such as promoters, enhancers, and other distal regulatory elements that might be controlling those genes? So a big shout out to the gene regulation community that has over many years developed these techniques. And ENCODE data are very cell type specific, which is both an advantage and a drawback to doing this. It's a drawback because you need to collect the data in many different cell phase and cell states, unlike the first approximation with DNA sequencing. But then it gives you richer information because the answers that you get back tell you something about what cell types are involved. And if you look at differentiation over time, total-potent cells start off with one set of candidate regulatory elements. Some of those are extinguished over time. New elements appear to be turned on in progenitor cells, and then other elements turn on in fully differentiated cells. So these changes in regulatory element signatures tell us about cell fate. So to date, ENCODE has done a lot of useful things and helped out the community. We're sharing thousands of data sets. Today they're shared without any embargo. There's no wait 1 year until the consortium has had a chance to publish on it today. They're shared through unrestricted access, the equivalent of GEO. There's no signing up for DDGAP or buying an account or something to access the data. The data are uniformly processed to a large extent, which minimizes the chance of coming up with cross-data set comparison and finding differential effects that aren't real or analysis artifacts. We're also sharing the software that we've either produced or used in the project so others can use it and also to make things transparent. We're working with other projects, other individual investigators to make the data more interoperability, talking about standards and how things are communicated. One of the things that we share our ENCODE publications, and there are hundreds that have come from the consortium, but perhaps for this audience more important, there are over 1,000 publications from outside of ENCODE that have used ENCODE data. Some of these look at human disease, some of these look at basic biology, and they cover a wide spectrum of diseases. And I think that this attests to the translational value of the resource. So in summary, we're trying to create a catalog of all functional elements of the genome. This is freely shared with the community. The foundation for this is established techniques and methodologies that have been used for years in gene regulation studies. And these maps can be used to make predictions about genome function. This can help you know the book. So now we're going to take a peek at how you annotations can't be used as one line mapping strategy. You've also got the issue that it's possible that multiple variants work on the same trait or disorder. You might not be trying to find the causal variant, rather a group of them. So ENCODE data can help you to figure that out. And a recently published study looked at ENCODE and Common Fund or Roadmap epigenomics data and found that many GWAS findings overlap with these annotations. So that suggests that this could be used. If one is not familiar with these kinds of assays, the simplest way to get started is there are a couple of pre-computed resources. First, I'll tell you about HaploRank from Manolis Pellis' group at MIT that can help you to sort through Again, you can put in genomic coordinates or step IDs, and then click Move On. And initially, you're presented with a score, the likelihood that the genomic region you're looking at has regulatory potential. And then, of course, you can drill down and see the individual underlying data and see why that assessment came about. As Gene said at the beginning, and I meant to say at the beginning but forgot, please don't worry about writing down URLs or detailed explanations. All of these slides are going to be shared. So that information will be available to everybody. There's also an ENCODE SysElement browser, and this has a number of different functions for human data and mouse data. And in this example, you can again put in a genomic coordinate, and it will find candidate regulatory elements. In this case, we learned about looking for DNA hypersensitive sites, and also tell you about candidate upstream regulators by looking for motifs that are found in those regions. And prediction of target genes is also an interesting problem. GTEx and ENCODE data and other projects are finding that it's not unusual that regulatory sites don't work on the nearest gene. They work on some gene that's further away. So if one finds a piece of DNA that you think is important and it's not protein coding, if you just guess that the nearest gene is its target, the growing story is you'll be wrong at least half the time. Okay. And if we look at, for instance, predictions based on GWAS studies, a lot of times it's quite distant that the regulatory region is from the gene. And in a practical sense, what this could mean is you might have some variant of interest and start studying the nearest gene, but perhaps there's a better candidate that one could work with, a better prediction. And that could change your research project if you didn't have to spend a few months studying one gene, studying the next where your only filter was proximity. If you knew other principled ways to say what was the likely candidate, you could come up with a better answer. So you can do this by using regulatory elements database. Terry Fury's lab developed this, and it works a number of different ways. One of the things you can do is put in genomic coordinates, and it'll again report DNA hypersensitive sites in that region, candidate regulatory regions. And if you click on any of them, it tells you additional information. And one of the things it will tell you is if there's a prediction for what gene is the likely target of that, it will give you a confidence estimate and what that target gene might be. The ENCODE SysElement browser, which I pointed out before, and in the bottom of these slides, you can see that you're going to be hearing more about this at workshop session 3, for example. So this is just a very quick overview, but again, you can put in a gene name, and this tool will return to you what are regulatory elements that appear, what are candidate regulatory elements that might be linked to this gene. Okay, and the one that I've highlighted here is one that I showed in that initial example, showing a common control region that affected many genes. I chose that region, that example, because in mouse that's been very well worked out. That region's been knocked out, and it does in fact control these genes. If you go to ENCODE human data, you could make the same prediction without having known any of them. And the last use case I'll tell you about is predicting target cell types. Again, to some people this is surprising that this is something you would need to do, to others it's intuitive. But if you think about most human diseases affect several cell types. Sometimes they affect several cell types directly, sometimes it's more subtle. For instance, sometimes the affected cell type is a developmental niche for the cell type that actually shows the dramatic effect. There are people who have lack of lymphocytes because their thymus, the niche where lymphocytes develop, is impaired. Right? So the affected cell type is not where that gene appears. And sometimes, of course, the etiology of diseases is not well understood. I'm old enough to be able to remember when it was a wild and crazy idea that type 1 diabetes, a metabolic disorder, might involve the immune system. And we now know it's an autoimmune disorder, but for some fraction of diseases that we work on today, we don't know the cell types. So again, in regulatory elements database, if you call up different elements, in some cases, those elements appear to be active in only some cell types. Other elements appear to be broadly utilized. So that could give you some information about cell type that's affected. Similarly, if you look at haploreg and regulome DB, which I talked about briefly, when you see the underlying evidence, if for instance you mouse over something in haploreg, it tells you the cell types that data came from. And that could guide your hypothesis, especially if something's known about the disease. You think Alzheimer's, okay, neurons, macrophages, glial cells, interface from glial cells, right? That helps you to refine things. It's more difficult to use if nothing is known about the disease. And if we look in the the ENCODE syselement browser and, for instance, look at gene expression, you can type in a mouse or human gene name. It might be the gene that you think is important in that disease is expressed in a restricted number of cell types. Obviously, some other genes are broadly expressed. And that, again, could guide your hypothesis as to what's going on. A more principled way to do this is you could intersect genetic variants that are linked to the disorder, intersect them with regulatory elements, and then ask whether those regulatory elements are enriched in particular cell types. And the code for doing this is shared at this URL on the slide, and then you can ask in a more principled way which of several candidate cell types might be involved in a particular disorder or process. So in summary, I think the main thing that ENCODE can help with is hypothesis generation and refinement, and that this can be used to predict causal variants, to predict target genes, to predict cell types and upstream regulators. All of the examples I showed you are examples of how you could use this with genetic results. If you had epigenetic results or an epigenetic cohort, you could do many of the same things with ENCODE data. Of course, if you're skilled in that, then you know that some additional caveats apply. The same kind of thing I've shown germline examples, but one could do all of this with somatic variants, but again, additional caveats apply when you have that. So now I'm going to quickly run through how you can access ENCODE materials. You're going to hear a lot more about this, at least in workshops 2, 1, and 5. So the ENCODE portal, encodeproject.org. That's the one URL I would want you to remember, encodeproject.org. It has a lot of this info. And for instance, you can access the data under either search or matrix. If you go to matrix, there's a nice representation, which I've crudely trimmed so you can't see most of it. It shows you the kind of data that's offered, data types, projects that the data come from, and so forth. And you can use facets to narrow down the subset of the data that you want to see. I want to emphasize the data policy. As I said in the beginning, this is freely shared. So if the data are there, you can use it. There's no restriction, no embargo or anything. We do ask a standard scientific thing that cite the project, if you, like with any other data source, if you use something cited. For reasons of transparency and reproducibility, it's good to share accession numbers. If you share the accession numbers of what you did, then your peers can tell what you did and how you did it. Okay. New to the ENCODE portal is the search by region. You can put in a genomic coordinate or SNP ID, find ENCODE data that overlaps with that region, and visualize it. That's a quick way for those of you that want to see tracks or to download elements to get your data. There's an ENCODE encyclopedia. You're going to be hearing about this. This is the focus of workshop 1. All right, and it has high level and very close to the data analysis, has a range of things. Ground level, close to the data annotations are things like where are transcription factors bound. Middle level annotations are putting the data together to find things like where are promoters and enhancers and how can you visualize them. And then top level annotations put together many different types of data to generate inferences. You'll be hearing about that especially in workshop 5. ENCODE publications. Again, there are community publications that I would point out to you because they're examples of how people outside of ENCODE are using the data to study human disease, to study basic biology, to produce software tools. There are also ENCODE print publications that, again, illustrate what was done and how the data can be used. We're very interested in sharing the standards for how things are done, and this is done again for the same two reasons: for transparency, so that you know what was done and how it was done, so you can evaluate for yourself how credible this is. And then we're also sharing these standards because there are many people in the field that are, including some that are newer in the field, and they're wondering, how should I be doing this? We hold this out as one way that you can do it, but of course it's for each group to decide how to do it. We don't dictate standards to anybody. So you can click through and see different types of standards and experiment documents that we share. We're sharing software that we're using in the project, again, so that people can understand what was done and how it was done, or if people are doing this kind of work and want to use software. Some of this software was created by ENCODE, some of the software was created outside of the project, but we use it heavily, so they're both important to know about. We share both of those kinds of software. And lastly, I'd like to give a shout out to our partners in the International Human Epigenome Consortium. ENCODE is a member of this. This is the NIH Common Fund Project Roadmap Epigenomics. A bunch of like-minded projects are collecting epigenomic data across many cell types and sharing them through different mechanisms. The ITAC data portal and project summary are available. There's lots of useful data there, including a subset of the ENCODE data. So I want to finalize here with a list of URLs that I don't expect you to read now or write down now. Again, all of these slides are shared. You click on the links rather than write them down, but we share, for instance, tutorials from the project, including the results of this will get posted online, analysis tools in the portal, and then I have resources. I highlight the ENCODE mailing list. If you want to hear regular updates on the project, you can join this list. So I'll conclude by reminding you that the goals of ENCODE are to find all of the candidate functional regions in the genome. This is an aspirational goal. I think there's no way to know when it's done or how it's done. And we're freely sharing the results that we find as a resource for the community. And ENCODE data are already being used in studies of human disease and human biology. And it's used any way that anyone can come up with. I'll stop here by thanking the people on the ENCODE consortium first. There's another representation of some of them. I have the privilege of working with smart people every day and hearing great ideas. It makes my job really fun. And my program colleagues at NHGRI, Elise and Peter Good, started the ENCODE project long before I had anything to do with it. Dan Gilchrist and I work on the project now along with Elise. And Dan is here today. I encourage you to say hi to Dan as well. And they're great colleagues and people to work with. And of course, all of this work, like any other bit of science, is built on years of study that came before it. Lots of work on gene regulation, lots of work on genetic basis of human disease. So I'll stop there and take any questions if anybody has any questions, or we can move on to the next slide. Okay, so now what I'd like to do is introduce our first scientific speaker, our keynote speaker. Oh, I have one question. Thank you.
So regarding the ENCODE feature, so what the NHGRI plans to do? Like, what kind of focus or emphasize?
Sure. Some of this is public record, and some of this is to be decided yet, and I can tell you about the public record part. And we've recently accepted applications for a new round of ENCODE. They haven't been reviewed and funding decisions haven't been made, but the applications we accepted were for new mapping centers, sort of like the production centers that we have now. Computational analysis centers, that's an ongoing activity that we have now in ENCODE. And then a new type of activity, functional characterization centers. We also have applications for those, in addition to a data coordinating center and a data analysis center. So there's a good chance that moving forward, there'll be more data production in ENCODE, more mapping. There'll be releases of data through the DCC and analysis, and then we might have additional information about functional different genetic regions. Past iterations of ENCODE did not focus on functional analysis of the mapping results. The thought was that was within the purview of the community. The idea was to do the mapping and to come up with raw material for others to work on. But we've seen that this is a need for us as well to be involved in, so that's why that's coming.
Thank you.
Sure. To our keynote speaker, we're honored to have Nancy Hux here from Vanderbilt, and she's going to tell us about her recent work looking at genetic contributions to gene regulation and how learning about that can help us to understand about the role of genetics in human traits and human disorders. So please join me in welcoming Nancy. Thanks.
Thanks. It's a real pleasure to be here. This is such a great meeting, and I've been really looking forward to sharing some of the newest findings. But I really want to get some feedback from an audience like this one and more collaborations with my ENCODE colleagues, because while this project was very much grounded in and started by research that we were doing in GSEC, it is very much going to end up in deeply looking at ENCODE. So what I'm going to do today, so I think about a year ago, I was talking about initial results in about 5,000 subjects from BioView. What I'm going to talk about today are results of studies that we've been doing with our PredictScan methodology in about 20,000 BioView subjects on our way to about 120,000. So there'll be some novel gene to phenotype discoveries and more on the continuum from Mendelian to common disease in a number of different dimensions. Some new ways that I think we have to really get more kinds of insights into biological mechanisms of disease and new big picture biology that relates to the medical phenome as a concept in and of itself. And I'm going to try to convince you the only tail of the distribution you ever want to be in is this one, which is the tally of the number of transcript levels.
RXR as a partner. Now, when we look at the RXR system, was actually much larger than that for the PDR. But the important point here was when we look at co-localization of the two, about 60% of the vitamin D receptor sites co-localized with RXR, suggesting, of course, that was supporting the idea that, in fact, this was really a primary function of the receptor was to utilize RXR as a partner. And when we looked at the response elements, we looked at a de novo examination of these binding sites. The most common element that turned up was an AGT-TCA duplex separated by 3 base pairs. And this actually was pretty satisfying because this was consistent with exactly the sequence we identified in the first gene, and I won't tell you how many decades ago that was. But in any event, these were then features that were consistent and validated those earlier studies. But as has been talked about today already, and was a surprise to us, we learned that, in fact, that most of the binding sites were not located promoter proximal, but were in fact intronic and intergenic, and could be 10 to 100 kilobases away from the promoter itself. And this has major implications, has already been discussed, in actually now identifying the sites of action of transcription factor, because this is a general feature, and the target genes are regulated. This has actually created a big problem for everyone in the room who's studying gene regulation. Now, the final thing is when we look at the osteoblasts, mature mineralizing osteoblasts, what you can see here is that incredible downregulation of system-4 vitamin D receptor, going from around 7,000 binding sites down to about a little bit less than 10,000. And this was actually a rather surprising statistic. So we were curious about whether there's a consequence to this, and so we actually did some microarrays and some sequencing analysis of the precursors in the mature osteoblasts, and you can see that there's a striking decrease in the number of genes that are targeted from about 1,000 down to maybe around 400 or so. So there was incredible downregulation distributed both in genes that were upregulated as well as downregulated. Now, this actually wasn't entirely a surprise because we know that 125D3 is actually trophic for the vitamin D receptor, and therefore in the absence of 125D3, the receptor actually decreases its expression over time as these cells actually mature. So this wasn't an incredible surprise, but what was a surprise actually was that we found a collection of genes, actually a very large set of those 400 genes, which showed differential target gene responsiveness to 125D3 that was entirely due to differentiation. And here's some examples here of the comparison of expression and response to 125D3, and then the data tracks, the CYP tracks for those individual genes across the site. So the first one would be called 2A1, which you can see is downregulated in precursor cells, but is relatively resistant to 125 action when the cells are actually mature. And I think it's not surprising. If you look at the track down here, there's a very strong vitamin D receptor binding peak right here. And in fact, that's almost completely obliterated when the cells mature. So this pretty much accounts for at least some of the segment of genes that are downregulated, that are lost when responsiveness is lost. However, if you look at the NTP1 and 3, these are mineralizing regulators involved in the osteoblast ability to mineralize their matrix. You can see that, in fact, their responsiveness to vitamin D over time is actually increased during the differentiation process. And if you look at this locus, these are genes that are actually separated or closely spaced, and in fact are regulated by the same enhancer. But the surprise is that even though these are increased in responsiveness to the vitamin D hormone, the amount of vitamin D receptor that's bound here is decreased by at least tenfold or more, which was a surprise. Finally, if you look at IGF-DP5, you can see it's not responsive in earlier cells and then becomes responsive to vitamin D as the cells mature. And again, in the same way, we lost a lot of vitamin D receptor binding. This is the scale over here, so we lost a lot of vitamin D receptor binding. And the real question here is how we sensitize these cells to the effects of 125D3 for the receptors. Now, dramatically downregulated, hence binding activity is not nearly as extensive. And the two answers to this are not simple, but one is epigenetic and the other is transcription factor activity separate from that of the vitamin D receptor itself. So I don't have time to go through the epigenetic changes that are involved in differentiation. These two descriptors here are really things that are one of the great contributions of the ENCODE project. That are localized to
we noticed, in fact, that if one looked at
we have to hint that two master regulators of the osteoblast, RUNX2 and CEBP beta, might be bound to those sites that the vitamin D receptor partner RSR. We're bound to. In fact, it turned out when we did a statistic analysis, you see about 40% of the vitamin D receptor binding sites actually have both RUNX2 and CEBP beta, and the binding sites actually correlate quite well. And in fact, what was most interesting about this was when we really looked at this, there was a definite organization of these binding sites where the vitamin D receptor was in the center, and RUNX2 and CEBP beta were on either side of the binding activity for these. We call this a consolidated enhancer, and we gave it the name osteoblast enhancer complex. I'm sure that it probably works in other cell types besides the osteoblast, but certainly there was an organization here. At a specific enhancer, there was other proteins that were involved in potentiating or controlling the regulatory capability of the vitamin D receptor itself, and we think that that's not unexpected and certainly interesting. So key features of these enhancers thus far, distal binding site locations. You're all pretty familiar with this, modular features, where there are multiple transcription factors can be bound. Epigenetic enhancer signatures can be seen in these sites, and in fact, the most interesting is transcription factor systems are highly dynamic, not only in the differentiation, which I showed you, but clearly, I'm sure there'll be others that will be talking about this, in differentiation, maturation, and disease activation, and they have a major consequence on gene expression. So I want to touch on now just two more genes that highlight some of the additional features of enhancers that we discovered. The first one is MMP13, which is regulated by vitamin D and during differentiation. It produces collagenase 3, which degrades cellular collagens, has skeletal sites in bone, but it also has very profound effects in cancer and in fibrosis and a variety of other areas. It's regulated by many things besides vitamin D, and the interesting thing about this is that all previous work on this gene has focused exclusively on the promoter proximal region of MMP13, and this is true of so many genes now that when we begin to study a gene, we try to ignore all the previous data that's been generated by standard luciferase assays and transcription analysis because it's almost always wrong. So you can see, and we contribute a little bit of that, although most of our stuff turned out to be not unreasonable, but I like to say it's either 5% right or completely wrong. But in any event, you can see here, this is another gene in that collection where there's really very little regulation in the precursor cells, but when you mature the cells, there's an increased sensitivity to 125D3. So here's the statistic analysis that looks at this locus. Here's the MMP13 in this direction, and here's the promoter region. And what we're looking at is both precursor cells and osteoblastic cells, and we're looking at a variety of different measures. I think the first thing you can see that is that in addition to the promoter itself, there are three different regions here, 10 kilobase, 20 kilobase, and 30 kilobase, that suggest that they might be enhancers. And interestingly, this one binds the vitamin D receptor, the 10K binds the vitamin D receptor, the 20K tends to bind CEBP beta, and the more distal one, the 30K, seems to bind largely RUNX2, which is the master regulator. And so we were curious about this. If you look at these sites, they all align with circular base 3K4 monomethylation. But you can also see some examples here where there's a difference between the precursor cells and the mature osteoblast, suggesting that there may be some differential activity that correlates with the expression as well, particularly down here with H36 methylation. You can also see some valleys here that John Stan has highlighted, and others have highlighted, where the binding activities actually are lost within the valleys that are associated with this. So to actually explore this in more detail, we used CRISPR analysis to CRISPR-deletion gene editing methods. In this particular case, we took these cells, parental cells, and we made a variety of deletions in CRISPR. And these are the deletions that we made. We did delete a region around proximal to the promoter, but not including the promoter. We deleted the 10 kilobase region as well as the 30 kilobase region. We also knocked out the DDR, and we also knocked out RUNX2. So we had a collection of daughter cell lines, and in addition to that, the beauty of CRISPR is that you can make sequential deletions, so that you can examine the combinations, and we did that as well. And then we looked at these daughter cells for the activity of MMP13 exfoliated in response to 125. Now these are 125D3. So these are the data in these individual daughter cell lines. This is the parental cell line here, basal, and 125 inducible. This is just a clonal cell line. This is another control. Now I'm not going to take you through all this because it's relatively complicated here. This is the general activity. We just expanded the axis in this on this side. But what I want to show you now is that, in fact, when you delete the promoter proximal region right here, you do lose some basal activity of MMP13 expression, but you retain to a large extent the 125D3 response at induction. If you remove the 10K enhancer, you'll see here that you also get a reduction in the baseline expression of MMP13 transcripts. But interestingly, you lose the induction, as might be expected, you lose the induction by 125D3, which you can see here is go down. But in fact, now you get actually a suppression. And I'm going to talk about this in the next slide, just to comment on what our hypothesis is. Finally, if you look at the 30K deletion, you can see you lose all basal activity and almost all the 125D3 responsiveness. And if you look at the transcription factor knockouts, you get similar correlations. So these were pretty interesting to us, and these data, along with some other data that I don't have time to show, led to this kind of not unexpected constant interaction model, where we centered the 30 kilobase of cancer, and we have aligned it with the 20 kilobase with 10 kilobase with the promoter proximal region, and largely because of 30K enhancer impacts, which binds RUNX, this master regulator, but not a binary factor, influences the activities of these other enhancer regions, and they in turn determine the output of the gene. And one of the additional experiments that we did was simply to see if we deleted the 30 kilobase enhancer, what happened to vitamin D receptor binding and RUNX binding to some of these other sites, and CEBP beta binding activity. And it was almost completely obliterated when we removed this distal 30 kilobase enhancer. So we think that this is very central, and we also would suggest that this could be very complicated. For example, if you found a SNP in this region, in this 30K enhancer, that would affect the binding activity of RUNX, and therefore impact the ability of this gene to be regulated by vitamin D. But you might search long and hard to find a vitamin D responsive region in this 30K element because it's not there. It's over here. And so I think this has an impact on these ideas. Finally, just to address the question, the 125D3 actually downregulates RUNX expression. And the dependency of this gene on RUNX is extremely important. And so if you downregulate the RUNX by a secondary activity or a separate activity by 125D3, we think that leads to something that looks like suppression.
Now, I'm going to turn to this last gene, rencligand gene, and I'll go quickly through this. Rencligand is an osteoclastogenic factor that we know is involved in bone remodeling and certainly involved in osteoporosis, but it's also involved in immune function. It's also involved in smooth muscle activity, and we think it's involved in atherosclerosis as well. And so we were interested in that. This is the gene, and you can see immediately we've looked at a variety of cancer regions upstream of this particular gene. You can see there's at least 10 different enhancers. These are the binding activities of various factors that we've looked at. I won't go through this in detail, but the key elements here are that these individual enhancers, here's the gene itself, actually, TNF-FF11. You can see that these enhancers actually are mediating largely mesenchymal or osteoblast lineage type cells. And these actually are regulating the expression of rencligand in T cells and B cells. So we went in and knocked out these 5 different cancer regions, and we've looked at the phenotype of expression of rencligand in these cells. And I won't go through all the details of this because it's rather extensive, but the reality is that what we were able to learn from these is they have individual temporal, hormonal, and tissue-specific activities that are very important. So just in the last 3 slides, I'll go through this very quickly. We've done a lot of other work, but we're actually interested in the impact of rencligand in the atherosclerotic plaques. And what we did then was cross the enhancer-deleted mouse, the D5 enhancer-deleted mouse, into the ApoE mouse, treated these animals then with a high-fat diet and looked at some of the features of this animal, phenotypic features at 12 and 18 weeks. And the first thing you'll see is that rencligand is regulated in the atherosclerotic plaque, but when you cross it with the D5, you can see a complete downregulation. Bone is effectively controlled in these animals, and we get an induction of osteopetrosis due to a downregulation of rencligand. So when we looked at the atherosclerotic